Good evening, everyone, and I'm Rishabh. And today we'll be starting with the discussion of week 11 of the course Experimental Biotechnology. In this week, the things that the lecture contains sequencing techniques, both DNA and protein, as well as the blotting techniques of RNA, that is northern blotting, and the DNA blotting, that is the southern blotting. We'll be discussing about the technique and some of the research problems that come into the field. Okay, let's start. For, we'll start with the DNA sequencing. What is the purpose of dideoxynucleotides in Sanger sequencing? The options are to elongate the DNA strand during synthesis, to initiate DNA synthesis, to terminate DNA synthesis at specific positions, or to label the DNA fragments for discussion. What can be the correct answer? Okay. Yes, the correct answer is option number three, and here is the explanation. Particularly, Sanger sequencing was the first technique, not the first technique. Uh, it's the technique that uses PCR amplification as a method to sequence the DNA. Okay, so for that, what we need is we need a single stranded DNA sequence that needs to be sequenced, and which will act as a template. Okay, so this will act as a template. Then, as we are doing a normal PCR kind of thing, then for that we will need a DNA polymerase that would allow us to get a multiple fragments of that particular DNA, and then we need the DNTPs, that is the deoxynucleotide triphosphates, for the particular DNA. Then we need the radioactively labeled primers that will help us to visualize the DNA fragments formed after the DNA replication. And then we need the most important component of the sequencing, that is the DDATP, that is dideoxynucleotide triphosphates. Okay, so as you might know that in a normal DNA sequence, DNTP structure, so this is the first carbon, this is the second carbon, third carbon, fourth carbon, and it's attached to the fifth carbon. To the fifth carbon, the phosphate group is attached. That's why it is called as the 5' end. And to the first carbon attached the base. Okay. And to the third carbon, that is the 3' OH, it's the chain continuation step. And at 2' position, we don't have a OH, instead we have a H only. This is a normal DNTP basic structure. And as the DNA replication progresses, it progresses in 5' to 3' direction. So the 3' OH is a necessity for the replication to be continued. In case of DDNTPs, this OH is replaced by an H that makes the DDNTPs. So that is 3' 2' dideoxy. So at both the 2' as well as the 3' end, the oxygen has been removed and it has been replaced by a hydrogen. So that is why it is called as a DDNTP. So whenever in a growing chain, for example, if we are doing a replication process in vitro, this is the primer, and if here, for example, if it's a C, then at normal position it will come as a G. That will be a DGTP. But if in then it can continue to replicate because here the 3' OH is present for the further replication process. But if in presence of this DGTP, here comes DDGP. Now, in that case, what will happen? The 3' OH is not present. So, it will not be able to continue the replication process, and here the strand will terminate. So this will lead to if we denature this DNA, it will lead to formation of two strands. Just a second. So it will uh, if we denature this DNA, it will lead to the formation of two strands, one with has the longer uh, length and one with has the shorter strand, because here the chain has been terminated, and this will lead to the formation of a smaller band. Okay, now this technique is exploited here, and so this technique has been exploited here. And here what we do is, after adding these reagents in a single tube, we divide this particular reaction mixture into four tubes that contains different DD NTPs. So, for example, we have DD-ATP, DD-CTP, DD-TTP, and DD-GTP. And these DD-NTPs are added in a ratio that is 1 in 100. Okay, that means for each, like for 100 DNTPs, there is one DDNTPs. Okay, why is this so? Because if we are having a chain termination, that means we will have like less number of chain terminations. So that will help us to yield a band on a agarose gel. Okay, so that is why we have used this kind of ratio for the particular experiment. Now, after incubating this with the dideoxy method, we run it on a agarose gel in different wells. So now what will happen, if we are running in different wells, then the chain that has been terminated at that particular position where it needs to be incorporated, so it will form a band there. Okay. Likewise, for every position at which A needs to be incorporated into the growing chain, that will form a band at that particular position. Okay, so same way for the other DDNTPs also. Now, what will happen? We'll read the sequence in this way, because this is the 5 prime to 3 prime reaction. The primer would be in this direction. Okay, so first band that will be formed, that would be smaller. So this would go here, and then it will move to the upper place. Okay, so we'll be reading the sequence from this direction. Okay, and this sequence that we will get of the this particular strand, that will be the complementary sequence, because remember that this is an extrapolation of the PCR technique. So the strand that we'll get is the sequence of the complementary strand of the original DNA strand. That sequence need to be identified. So now this sequence would be the complementary sequence, and this sequence, when we'll deduce the complementary of this would be the actual DNA sequence that we have already sequenced. Okay, so this is the final sequence of the original DNA strand that we are looking for. So this was the original technique that was used by samplers. Now the modifications of this technique has been used, and what do we use is, instead of a radioactively labeled primer, because this used radioactivity, so here the most of the steps are like increased because of that. So now the modified step uses fluorescently labeled DNTPs that are fluorescently labeled. So different DNTP, for example, DNTP is labeled in green, DCTP is labeled in red. So they are labeled in different colors. So now, if we are, and likewise, we run this particular thing in a capillary instead of a agarose gel, we run it in a capillary. So whenever the thing is incorporated, for example, if DATP is DDATP is incorporated here, so in that case, what will happen is will that this will show a red color, and that will be detected by the system or the instrument. Likewise, for every other DD and TP, different color would be there. That will be detected by the system, and that would give us the complementary sequence of the original DNA strand. Okay, so this is the modification, and this has been used in a high throughput DNA sequencing techniques nowadays. Okay, so any doubts in this particular method? Okay, if not, then we'll move on to the next question. Which component of Sanger sequencing reaction allows for the visualization of the separated DNA fragments? What can be the correct answer? Okay, any more answers? Okay, so as I have explained just now, the initial technique used radioactive primers for visualization, and the newer techniques that we are using are using fluorescently labeled DNTPs. Okay, so according to me, both are correct answers, second and fourth. Okay, so this would be second, because in the initial days, the radioactive primers were used, and autoradiography was done, and now we have the fluorescently labeled TDNTPs. So both the options are correct. Okay, so any doubts? Okay, if not, then we'll move on to the next question. Yeah, so this is a similar explanation. If we are using fluorescently labeled DNTPs, so in that case, that would give us peaks like this. Okay, so this is how the data is being visualized in nowadays as the high throughput sequencing techniques. Okay, so the next question is, in Sanger's method, the fragment of DNA should be first dashed before proceeding for sequencing. What can be the correct answer? Okay, any more answers? Yes. So the correct answer is option number three. So as you have looked in the previous slides, so first that the template we have, sorry, the template we have needs to be PCR amplified, and then it is basically subjected to the sequencing using various primers, the NTPs. Then they are separated into different columns with DDNTPs. Okay, so let's move on to the next question. So this is an analysis based question. You have a given DNA fragment that have this particular sequence, and these X's and the Y's that are present, these are the primer binding sites. So what are the sets of fragment that you will get out of this particular DNA sequence that is given? So take your time and choose the correct option. Okay, that's quick. Any more answers? Okay. So when the primers will bind, they will bind in this direction. I have not written those primer binding sites here. Now, whenever the bands will be formed, it will be formed in this direction. Okay. So now we'll check how the bands will be formed. Now, first, if the DDCTP gets attached to it, it will form the first band. Then comes the GTP, DDGT. Then this one, G again. Okay, so this was the final fragment. So we'll check for the options. So we will have CG. Okay, G would not come first here. So both first and the fourth option gets excluded. Now we have the CG. Okay, so this fragment. Then we have CGA. So CGC cannot come. Okay, so this option is also neglected. Now we have the CGA TCG. So CGA TCG. This is the second last fragment. This one, and then we have CGA TCGC. So this was the final fragment. So yes, the correct answer is option number three. So any doubts in this question? Okay. Okay. So let's move on to the next question. So here is another analysis based question. What will be the sequence of original DNA from the given DNA gel? So here the gel is given from the Sanger sequencing. You have to deduce the DNA sequence of the original DNA. What can be the correct answer? Okay. Any more answers? Okay. So the correct answer is option number three. Okay. So let's see. So from here, the DNA sequence that will come will be T A C C T T G A T 3. Now, the original strand would be A T C A A G G T A 3. Okay. So the correct answer would be option number one. Okay, not three, not two. Correct answer would be option number one because this would be the original sequence, right? So that is why this would be the sequence of the DNA that is being given by the sequencing. So this would be the original DNA sequence. So any doubts? Okay. So let's move on to the next sequencing method, that is the Maxim Gilbert method. Oh, sorry, there is one more question. So that is related to the, so using Sanger's deoxy chain termination method, a particular exonic region of a protein coding gene was sequenced for two individuals referred to as subject 1 and subject 2. The figure given below shows a segment of the autoradiograph corresponding a single window of the DNA sequence. Okay, so this is the gel that is given for a typical DNA sequence. Which one of the following interpretations is correct for the sequence DNA fragments? So there are the options. You analyze the data and then we'll discuss. Okay, so shall we discuss or you need more time? Okay, so this question is based upon the difference in the markers, genetic markers basically. Okay, so that makes every individual different. Okay, so what we are using is we are using Sanger's technique or sequence to detect these markers in different patients. Here we have the two patients, that two individuals, that is subject 1 and subject 2. Okay, so now first we will see the sequence from both the ends. Okay, so this would be A, then it would be G, again G, then C, then T, then again G, A, and then T. Now for subject 2, what will be? It's A, G, G, then C. Then at this position there can be A or T because both the bands are there. Okay, and then we would have the G, then A, and then T. Okay, so now what will happen if we see this? So starting from here, if we deduce this complementary strand, that would be A T C A G C C and then 3 prime. Okay, so subject 1 has a sequence that is A T C A G double C T. Okay, so this option is wrong. Option B is wrong. Then we'll see with the second subject. Okay, so this is also the original DNA sequence of the second subject, and this option is also wrong. Now comes the two options, that is A and D. So as you can see, if there would be a base deletion at any of these positions, then the sequence would vary. Okay, and it would shift from one of the bases to the other. For example, if there would be a deletion at this position, okay, so then this would, the C would shift here, and all the bases would shift one place. Okay, so this would be a frame shifting, and that would match to a certain extent to this particular subject. Okay, so that is not the case. So option D is also wrong. Now, as you can see in subject 2, both the probabilities are there that at this position both A and T can be present. Okay, so that means that it can be a allele variant of this particular gene, where in this individual T is present and in this individual both A and T can be present with equal probability. Okay, so these are called as allele variants, where a single base can be modified to the its other counterpart, and it might not change the protein sequence, or it can change the protein sequence. Okay, so this is the case. So option 1 is correct. Okay, and here it can also be the case of the heterozygous individuals. So I hope you guys know what is heterozygous. If not, then I can explain. So in case of heterozygous individuals, what will happen? As you all know, we have homologous chromosome, right? So there are two copies of the same chromosome that we have, one from the maternal and one from paternal. Okay. So uh, I think I spoke the opposite way. So this is paternal and this is maternal. Okay. So this would have slightly different variations if the allele have a different counter. For example, just for example, I'm taking for height. So for tall height, it's capital H, and for small height, it's small h. So this variant can be capital H, and this variant can be small h. Okay. So these are two allele variants that can be present in subject 2. That is why it is showing two bases at the same position. Okay. So any doubts? Okay. So give me 1 minute. Okay. So let's move on to the next question, that is related to the Maxim Gilbert sequencing method. So what are the typical order of steps in the Maxim Gilbert sequencing method? What can be the correct answer? Okay. Any more answers? Okay. So the correct answer is option number two, and here are the steps. Okay. So this was the first method that was basically discovered because this is a chemical sequencing based method. Okay. So now for this we would have a sample DNA that would be good in amount. Then we would separate this DNA, okay, and we will get a single stranded DNA. Then we add a 5 prime 32 at the 5 prime phosphate at the 5 prime end, because phosphate is present at the 5 prime end. This is basically radioactive labeling of the phosphate. Then we cleave these particular specific nucleotides at the sites, and then, sorry, first we chemically modify the nucleotides, and then we cleave this uh, DNA nucleotides, and after cleaving we yield different reactions, that is G, then A plus G, T plus C, and C. So these are based upon the different chemicals that we are using. So the reaction is terminated at these particular position or cleave. Okay. Based upon that, we'll get the bands wherever G will be present, the chemical would have cleaved that particular DNA and formed a band till there. Okay, because G has been first modified and then cut. So we'll get a band wherever G will be present. Then in the second case, where A plus G is present, that means the chemical is specifically recognizing the A and G. So wherever in the sequence A and G would be present, the band will be formed, both at A as well as at G position. Okay. Same is the case with T plus C. So in this case, we have the pyrimides only, that is T and C. So wherever the T and C will be present, that would form a band at that particular position, because that particular site has been cleaved. Then the final reaction is with C only. So here only the C will be cut and the band will be formed where C will be present. And then what we do, we run a Agarose gel electrophoresis and do a autoradiography. That will yield us the bands that has been formed at the particular locations where those particular sequences would be there. And As you know, in the electrophoresis, the farther it moves, the smallest size it would have. So this would read the actual sequence starting from the 5 prime end. Okay, so this would be the smallest band. So this would be the first to appear in the sequence. Then moving on with the each of the bases. Okay, so this is how the Maxim Gilbert sequence is done using different chemical methods. And so the option number two, that means first we have the DNA cleavage, followed by the chemical modification of the bases, then we run a gel electrophoresis followed by the visualization. Okay, so option number two is correct. Okay, so is it fine? Any doubts? Okay, so let's move on to the next question. So hydrogen in presence of NaCl opens up the. I know the answer is on the screen. So the correct answer is option number A, at the cytosine. Okay. So these are the different chemicals that we use for cleavage as well as modification. So first, the modified base is used. So there were four reactions: G, A plus G, T plus C, and only C. So for the G reaction, we use only dimethyl sulfate, that is DMS. Okay, so that modifies the N7 of the guanine base, and that opens up the ring. Okay, so now this would yield the piperidine to cleave at that particular location. Similarly, when G plus A reaction needs to be done, so we use a formic acid. So that basically attacks the glycosidic bond between the base and the sugar. Okay, then we have the reaction 3, that is hydrazine. So it breaks the ring of the pyrimides, that is TNC. That is specific for that. Now, if we have to only choose for C, then we have the hydrazine that and plus salt. So that specifically recognizes the cytosine ring and breaks it. And then after the chemical modifications, the phosphodiester bond is cleaved using the piperidine. Okay. So these are the four reactions and the four chemicals that we use. So any doubts? Okay. If not, then we'll move on to the next question. So you are given a agarose gel. Now you have to deduce the sequence of the DNA. Okay. And this is used in using the Maxim Gilbert method. So what can be the correct answer? Any more answers? Okay. So in case of Maxim Gilbert method, it is not the complementary sequence. So this would be the direct sequence that would be given. Okay. So it would starting with C. So in this case, if A plus G and G would have the band at same position, that means at that position G would be present. And if there is no band in the G column, that means at that position A is present. Same is the case with the C and C plus T. Okay, so let's reduce the sequence. So in the first base, it's C, then T, then it would be A, then G, then A, then T, then it would be A, then G, then T. Yeah, just a second. Then it would be A, then T, then again A, and then the last one is G. This would be three. Okay, so this would be the sequence of the original DNA strand of the patient or the person. Okay, so let's take the correct answer. So this is CTA, GATA, then GTA, TAG. So second option would be the correct answer. Okay, yeah, someone raised their hand. So do you have any doubt? Someone raised their hand. Okay, so let's move forward. If you have any doubt, we can discuss in the presentation or at the end. Okay, so let's move to the next question. So cleavage reaction in Maxim-Gilbert method is used to cleave the phosphodiester bond next to the modified nucleotide. It is done with the help of what was the chemical? Can you name now? Okay, any more answers? Okay, so here we are talking about cleaving the phosphodiester bond, right? So the cleavage is generally done by the hyperidine. So hydrazine with NaCl is used to break the ring of the cytosine residue only. Okay, and DMS is used for G and formic acid is used for A plus G. Okay, so hyperidine is used for breaking the phosphodiester bond. So the correct answer is option number two. Okay, any doubts? let's move on to the next question. Now we are moving towards the protein sequencing. So just now we have completed the DNA sequencing wala part. Now we are moving to the protein sequencing. What is the primary purpose of using the dithiotritiol in protein sequencing? What can be the correct answer? Okay. So the correct answer is option number three, that is to reduce the disulfide bonds in the protein. And here is the explanation. Okay. So what happens whenever we are doing a protein sequencing? As you all know, there are different uh, layers of protein structure. Okay. So these are the four different stages or the four levels of protein structure that we generally study. So the primary sequence is the amino acid sequence of the protein. Then it folds upon itself to yield the secondary structure that majorly involves the alpha helix or the beta sheets. With the help of the hydrogen bonds, they fold upon itself, forming different helices or sheets. Now, it has to fold upon itself. Okay. Now, it forms the tertiary structure that basically alpha helices and beta sheets, they fold upon themselves, forming stable bonds, and that forms the tertiary structure. Okay. Now, this tertiary structure with different subunit of proteins fold upon itself, forming a functional unit that is the quaternary structure. Okay. So the major active site of the enzyme or the functional part of the protein is formed by the tertiary structure. And if there are different subunits involved, then in that case it moves to the quaternary structure. Okay, so here the different subunits are involved. Now, what happens in the secondary structure? The main bond that is being used is hydrogen bond, and in the tertiary structure, major bond that is being used is the different van der Waals interactions. Here you see the hydrophobic interactions, hydrogen bonding, disulfide linkages, and the ionic interactions. So these are the major interactions that are being used for the protein to fold upon itself, and this forms a functional fold. Okay. So now, when we have to do a protein sequence, that means that we have to make the protein available in this particular sequence in a straight chain. Okay. So for that, we need to break all these interactions between the protein, hydrogen bonds, ionic bonds, and hydrophobic interactions break based upon the pH and the buffers that we are using. Okay. But these disulfide linkages are generally non-breakable because these are the covalent bond between the sulfide bonds. Okay. Generally, the cysteine residue forms the sulfide bond between the two cysteine residues. So we have to reduce it. And this reduction is done using dithiotritiol or beta mercaptoethanol. So these two are the reducing agents that are generally used for the denaturation of the proteins. Okay, and this is the typical structure as you can see here in the green. This is the typical structure of the dithiotritiol that is used for denaturing the proteins. Okay, so this helps in reduction of the disulfide bonds, and that would help us to open the chain. So this is the explanation. So any doubts? Okay, if not, then we move on to the next question. So cyanogen bromide cleaves the protein sequence at which position? What can be the correct answer? Yes, the correct answer is option number two, after methionine. So here is the table given for different reagents or the different proteins or enzymes that are used for breaking the bond of different different amino acids, and then we generally use the mass spec to reduce their size and further to reduce their sequence. Okay. So first is trypsin. So generally it is an enzyme that is present in our small intestine. Okay. So it cleaves after the positively charged amino acid, that is lysine and arginine. So that depends upon the amino acid that is present in its active site. Okay. So that's why it cleaves after lysine and arginine. Then after the chymotrypsin, it cleaves after the aromatic amino acids, that is phenylalanine, tryptophan, and tyrosine. Pepsin, generally present in our stomach, cleaves at the leucine, phenylalanine, tryptophan, or tyrosine. Okay, and cyanogen bromide cleaves after the methionine. Okay, so this is the answer. Any doubts? Okay, if not, then we move on to the next question. Which of the following is a Sanger's reagent? What can be the correct answer? Okay, any more answers? Okay, so the correct answer is option number one. Okay, and it was 1 fluoro 2,4 dinitrobenzene. Okay, here is the structure that is being shown of FDNB. So this interacts with the N terminal of the protein. So this is a protein with different amino acids and labeled in different colors, and this is the N terminal, free N terminal of the protein. So now this interacts with the fluorine group, interacts with the amino group of the protein and forms a complex, okay, that has attached to the N terminal of the protein. Now we have an acidic setting. So this acidic step helps us to release the modified amino acid, and further the amino acids are being separated out from the protein. Okay, so this technique was generally used to modify the N terminal, and then what we do is we run the HPLC or mass spec to reduce the different amino acid sequence that we have. Okay, and we will know what will be the amino acid that will be present at the end terminal. Okay, so this is how the Sanger sequencing for protein is done. And he sequenced the first protein, that is insulin. So it was generally the 51 amino acids that he sequenced alone using this reagent. Okay, so any doubts? Okay, so let's move on to the next question. So there is another method that is called as Edman degradation in protein sequencing. So what is the actual result of the Edman degradation method in the protein sequence? What can be the correct answer? Okay. So the correct answer is option number one. And what happens in the Edmonds reagent? So phenyl isothiocyanate is the actual reagent, Edmonds reagent that we call it. So PITC. This is the reagent that we use. So now using this, first it modifies the N terminal, the free amino acid that is at the N terminal. Okay, it modifies it and it forms in low pH. We can separate it out, the modified amino acid, and the rest of the ring or the rest of the peptide is separated out. So it is not degraded as in case of the Sanger sequencing method. So only the modified amino acid is separated from the rest of the protein. Okay, and then what we do is we determine what this amino acid is using different methods, and then this reaction is again continued. So this cycle is again continued with the other amino acids of the chain. So we have to again, like for example, this is the protein chain that we are talking about, and this was the first amino acid. So first PITC will uh, basically attack this, and this will be separated. Then secondly, this would be done. Then third and fourth and fifth. Likewise, for every amino acid, this cycle would be used, and it would be help us to determine which amino acid is which. Okay. Although it is time taking, but it will give us the efficient answer that what a protein sequence would be. Okay. So first step would be at high pH, and the subsequent steps would be at low pH because we have to separate the modified amino acid. Okay. So any doubts? Not, then we'll move on to the next question. What role does a carboxypeptidase enzyme play in protein sequencing? What can be the correct answer? Okay. So generally we use two kinds of peptidases in a protein sequence. One is an endopeptidase and one is an exopeptidase. Okay. So exo means from the last, that is exit. Okay. So for example, this is a protein chain. If the amino acid starts removing the amino acid from the terminals, that is an exopeptidase. And if the endopeptidase is cleaving in the middle, that is an endopeptidase. So in between. Okay. So the trypsin, chymotrypsin, pepsin are all endopeptidases because they cleave at the middle. And exopeptidases, for example, carboxypeptidase or aminopeptidase, are the exopeptidases. Okay. So carboxypeptidase are the enzyme that cleave the amino acid from the C terminal, and aminopeptidases are from the cleave the peptide from the N terminus. Okay. So here we are talking about the carboxypeptidase. So they cleave the protein from the C terminus. So they start removing the amino acid from the C terminus of the protein. So the correct answer is option number two. Okay. So any doubts? Okay. So let's move on to the next question. Now we are moving towards the northern block. So, which of the following is a common method for detecting RNA hybridization in northern blotting? What can be the correct answer? Okay. Any more answers? So, the correct answer is autoradiography. Okay, because we will be using radioactive labeled probes. Okay, so that is why autoradiography is the common method that we, if we are using fluorescently labeled probes, that is generally not used. So then in that case, microscopy, fluorescence microscopy can be used. Okay. So any doubts? Not, then we'll move on to the next question. Dash affinity column is used for purification of the mRNA. What can be the correct answer? Okay, any more answers? So, a eukaryotic mRNA have the following components. Okay, so it has a 5 prime cap, that is a G cap, followed by the uh, ribosome binding site, the exon sequence, okay, the continuous exon sequence, the poly A T. Okay, so this poly A tail is generally used to separate or purify the mRNA. Okay, so in that case, poly T will be used as a method to purify this particular thing. Okay, so the correct answer is option number 3. So a column, affinity column that is labeled with poly T will be able to purify the RNA that would have a poly A tail. So this would be the mRNA. Okay, so this is how the RNA is purified in RNA purification techniques. So any doubts? Okay, if not, then we'll move on to the next question. So, yes, this was the original sequence of the mRNA that I'll explain. Okay. So, what is the significance of pre-wetting the nitrocellulose membrane before mRNA transfer in the northern blotting? What can be the correct answer? Okay. Any more answers? So, basically, blotting. What is blotting? Okay. In scientific terms, it means that we are transferring anything on a membrane. Okay. So, In case of northern blotting, we are doing or separating a particular RNA on a blotting sheet. So what we do is, this is a gel, these are the bands, and that is transferred on a nitrocellulose membrane. Okay, and then we add the probe that detects the particular RNA that we are looking for. Okay, so now if this blotting sheet or the nitrocellulose membrane is pre-wetted. So why it is pre-wetted? So that the RNA that we are talking about basically moves to the nitrocellulose membrane. Okay, and it increases the binding of the RNA to the membrane. Okay, so that would basically what happens, because this is a capillary action based method. So many a times this RNA that is needs to be attached to this particular nitrocellulose membrane washes away. Okay, because this is, firstly it is delicate, and the second thing is, if the binding is not proper, then it will wash away. Okay, so that's why to increase the binding efficiency, the nitrocellulose membrane is pre-wetted. So the correct answer is option number 4. So any doubts? Okay, if not, then we move on to the next question. The northern blotting, the autoradiogram is developed at which temperature? What can be the correct answer? Okay, so the autogradiogram is developed at the minus 80 degrees Celsius. Okay, because this requires really cold temperature. So this generation is a heat generating step. So for that we require a cold temperature so that the RNA is also not degraded. And because RNA is really fragile, in normal isolation in the lab itself, sometimes it gets degraded without any reason. So we'll not be able to deduce why it happened. So to prevent all these things, the autoradiogram is generated at high, like very low temperature, so minus 80 degrees. Okay. So any doubts? If not, then we'll move on to the next question. After RNA transfer in northern blotting, what is typically done to fix the RNA on the membrane before probe hybridization? Okay. So the correct answer is option number one. Okay. So because we have to fix the RNA on the membrane. Okay. So alkaline treatment is generally done to remove its secondary structure. So that is generally done in the reducing page or the reducing agarose jelly electrophoresis. But to fix the RNA on the membrane, we use UV cross-linking because now, just I told you, the pre-wetting step. Okay, so that is one of the ways that how we can increase the efficiency of the transfer to the blot or the membrane. Now, the second thing is the UV cross-linking to let the RNA stick to the particular northern blot so that it doesn't washes away. Okay, so any doubts? Okay, if not, then we'll move on to the next question. What is the primary purpose of the southern blotting in molecular biology? Okay, so I think like both second and third uh, question options are correct because in general southern blotting is to determine the DNA fragment. Okay, in general, and if we are talking about the probes, then yes, it is to detect and analyze specific DNA sequences. So, in my opinion, both second and third would be the correct answer for the southern blotting. Okay, so any doubts? Okay, so let's move on to the next question. Which of the following is advantages of the vacuum transfer over capillary transfer in southern blotting? What can be the correct answer? Okay, any more answers? So yes, the correct answer is option one. So what happens in case of the vacuum transfer? It takes around 14 to 20 hours. Okay, to transfer the DNA from the agarose gel to the blood. And capillary transfer takes more time. So it takes around 24 to 30 hours. So to decrease the time of transfer and to maintain its nature, the vacuum transfer has more advantages over the capillary transfer. Okay, so this is the typical southern blotting technique that is shown here. Okay, so any doubts? Okay, so let's move on to the next question. Okay, so this leads to the end of our presentation. If you guys have any doubt in the presentation or in any other concept that you need to understand from the whole week, you can ask. Okay, if not, then we'll close the session for today. And thank you guys for joining. You can see the video in YouTube if you want. The recording of the lecture will be uploaded. Okay, thank you guys for joining. We'll close the session.